Hello everyone. My name is Najee Gaines, um, senior biology major, chem minor. This is my final bioinformatics PowerPoint presentation. And the PowerPoint presentation will actually be focusing on African American men with prostate cancer in the United States. So, the introduction of study. We're going to start off at the number one fact of it's a hereditary linkage. It is an ideal that portrays the structural planning of genomes and variety between populace. So basically, this is meaning in a sense that depending on location, we see a definite comparison between location of patients and individuals who have been diagnosed with prostate cancer who are also African American men. Next, we will start off and just say that all hereditary variability starts locally as another change independently and are geographically confirmed. There is a connection between the heredity variations found in one's geographic distinction in contrast with those who are discovered somewhere else. So that's what I was stating initially. The variations that are the normal crosswise over for humans populace have a tendency to be older and go back to the seasons of human development for about around 100,000 years prior. So that's important. And African Americans are an admixed populace, which implies that they have a late, I'm sorry, I'm a little sick, which implies that they have a late bling familiarity gatherings in this way for the illness that are disproportional, influencing the African American group and culture, essentially, obviously, in African American men. There may be attribute rarity variations that may be specific to only African American men to the increasing risk. Understanding into the dangers of loci, to explore is important as well. So this is just a small little snippet of what my R studio kind of represented, just for a quick little few touch bases. Obviously, the CHR's chromosome, we have, the SNP, that's the SNPs. We have age incorporated into the database, as well as the p-values. We have the OR, that is just, the odds that a mutation will occur given in a particular exposure. Obviously we have the p-value, which is the probability that a mutation will occur due to a specific change. And hopefully everyone knows what the SNPs are. They're just single nucleotide polymorphism. So that's just a small little overview of what the initial data look like. African diaspora, obviously the African people started off over in Africa, but it was exported through history as well as other migrational movements that African people, which turned into African American people, and etc., ended up in other places of the world. So it's definitely a factor of being incorporated into translocation, which is simply initially the key. Next, we're going to start off with the graphs that I've actually been doing in the R Studio. So the first graph is a MDS plot, and that was in the Geneva database, and that is a multi dimensional scale plot This graph basically clusters the groups of controls and populations that took action in a plink. The multidimensional scalar graph is basically the meaning of visualizing the levels of familiarity in individuals' causes of a data sheet. It refers to a set of related origins, technology bases used in information to visualize in a particular to display information contained in a distance matrix. So, and this is my MDS plot. I wish I could have gotten it in color so it could have actually seen what the actual like clear results. Obviously it would be nice diagnosis in color, but you can see that it's extremely popularly congested over in the left side of the region of the graph, and this was done through Geneva. So we're gonna continue on. So the next graph I did was the Manhattan plot, and that was actually in the Geneva as well. This is revolving around the chromosomes that were used in reference of the negative log of the p-value. So a Manhattan plot is actually just a type of a scattered plot graph that is usually used to display data which are in large amounts of data points that may include of non-zero amps, as well as distributes of high magnitude values. For instance, we have the genome-wide association study and other familiar studies as well. So this one I did was able to get in color. This is the Manhattan plot, and this was done in Geneva as well. As you can see, it's evenly distributed in some of the chromosomes ranging from 1 to 24, but you can see along the lines once you get to 15, 18, 22, the amount gets less and less as far as the color association between the orange and the blue. Another graph would be the Geneva significance test. And this test is just representing the individual's Q-value cutoffs, the significance test, as well as the p-values, how they're in comparison to each other. You'll see that when you look at the p-value, there's down in the bottom left corner, there is a gradual approach to growth, a slight, not too noticeable dip, and then a extremely amount increasing of acceleration. And then for the significance test, you see that, there is a gradual kind of even plane down at the bottom in the lower right corner. And then there's actually kind of like a jump of a acceleration. So the last graph will be the p-value histogram. And this graph is basically just letting you know the distribution of mutations within a specific p-value. The graph gets a immediate sense of how tests behave across all hypotheses, and it gives you an immediate diagnosis of some potential problems when you do run it when it comes to comparisons of different information. There were p-values found for all mutations, which results in clustering. So that is the where the density becomes clear afterwards, and the plotting, which then accordingly to relate to the different density levels, which are shown in the graph. And here's the graph right here. And this is actually the last graph that we did throughout our research, seeing how this research is actually going on right now at Clark Atlanta University in the biology department. There is no final synopsis on details when it comes to what exactly is going on in the final stages of the project. So it is continuing on now. So the data was just being used. From what I can see from the data, uh, there isn't too much of a clear understanding to why the prostate cancer is so prevalent in the men right now. There is commonalities when you do look at chromosome and certain mutations. So I do think that the research needs to be further, but overall, I think it was an awesome study. So thank you so much. I enjoyed your class, Dr. Quinn. It was great working under you, and I learned a lot. So goodbye.
That's too much background noise.